Hi everyone, I would like to report to you our study on the interaction between human interferon gamma and heparin or heparan sulfate oligosaccharides. Human interferon gamma is a signaling molecule with antiviral, antibacterial, immunomodulatory and anti-proliferative properties. It regulates over 200 genes and is clinically used in the management and treatment of various diseases. However, its activities are also associated with multiple autoimmune diseases and neurodegenerative diseases, as well as some cancers. So people are trying to develop inhibitors for its activity, but this requires proper understanding of its interactions and binding. Interferon gamma, shown here in red, is a homodimer which binds to two identical receptor molecules on the cell surface, here in green. The receptor cytokine binding interfaces here and here are shown in orange and blue bubbles. This is the crystallographic signaling complex, which lacks information about the positions of the highly flexible and negatively charged C-terminal tails of the interferon gamma homodimer. These C-terminal tails are extremely important because they affect dramatically the binding affinity of the cytokine to its cellular receptors. So we added them, reconstructed them, we folded them using long-scale molecular dynamic simulations, and this is the final structure that we use as input in our further studies. The C-termini here are shown in blue ribbon. Interferon gamma is also known to interact with the glucosaminoglycans heparin and heparan sulfate. These are polymers of uronic acid and glucosamine, which are highly sulfated and thus have a high negative charge density. We studied this interaction in two different cases. The first one is the free human interferon gamma molecule in the bloodstream, and the second one is the homodimer at the cell surface prior to its interaction with its receptor. In the first study, we used this hexasaccharide as a model structure for low molecular weight heparin. We placed 4 such chains in the vicinity of the C-terminus of human interferon gamma, and we found out that they all very quickly bind to the human interferon gamma molecule, and in about 200 nanoseconds all 4 chains are bound to the cytokine. The contacts are primarily located in the C-termini of the two human interferon gamma monomers here and here, and we also have some contacts with positively charged solvent-exposed domains in the globule. This complex between the cytokine and the 4 heparin chains has no longer a positive charge, but a negative net charge. This is the electrostatic potential surface of the cytokine, the full-length human interferon gamma free in a solvent. As you can see, we have a local positive local charge density at the bottom part of the globule, which compensates the negative charge density here in the receptor subunits. Once we have all heparin chains attached to the cytokine, the whole electrostatic potential surface is deformed and reversed, and this complex no longer can interact electrostatically favorably with the negatively charged human interferon gamma receptors. On the other hand, in the other study, we wanted to see how heparan sulfate oligosaccharides would affect the binding of interferon gamma to its receptor. So we used these 4 different scenarios. This is the reference simulation that we run, starting from the crystallographic complex. Then we have the full-length human interferon gamma separated from the receptors at a distance of about 4 to 5 nanometers, and we use the same input configuration with either hex hexa or octasaccharides in between the receptors to mimic the presence of heparan sulfate proteoglycans at the cell surface. We monitor the distance between the binding sites in the receptor and the cytokine at the two sites of the signaling complex. In the reference simulation, these distances had values of about 7 to 8 angstrom. As you can see, when we don't have glycosaminoglycans, the positively charged C-termini binds to negatively charged domains here in the receptor molecules and prevent the globule from contacting with the receptors properly, so no complex is formed. Once we add hexasaccharides in between the receptors, they attract the C-termini of the cytokine and pull down the whole globule of interferon gamma in between the receptors. However, only one of the contacting interfaces is formed properly. The other one is deformed and no binding occurs with the second receptor, as you can see here on the distance plots. If we add octasaccharides, which are experimentally known to bind with the highest affinity to human interferon gamma, these octasaccharides have higher net negative charge than the hexasaccharides, and they pull and engage the C-termini of the cytokine much more efficiently than the hexasaccharides, and they manage to pull the whole globule properly in between the receptors and provide for opportunity of the globule to properly orient itself relative to the receptor, so that we observe proper formation of the cytokine receptor complex. These are the contact maps in the final 150 nanoseconds of the simulation. So these simulations had a length of 600 nanoseconds. This is the reference contact map for one of the receptor chains and one of the monomers of the interferon gamma homodimer. As you can see, when we don't have GAGs, this chain does not cytokine chain does not interact with this receptor at all. Once we add hexasaccharides, we recover, at least qualitatively, the binding between the cytokine and its receptor, and when we add octasaccharides, we obtain a contact map which very closely resembles the reference contact map. We also tested our computational results experimentally. We inhibited the sulfation of heparan sulfate proteoglycans on wish cells using sodium fluoride, and we found that the activity of interferon gamma on these treated cells is reduced by 86% compared to the activity of untreated cells. So this is a qualitative validation of our in silico results. So in conclusion, glucosaminoglycans play dual roles in the interaction between interferon gamma and its cell surface receptor. On the one hand, once heparin comes across free human interferon gamma in the bloodstream, it binds to it and prevents further interaction of the cytokine with its receptor, so it can be used as an inhibitor of endogenous interferon gamma. But on the other hand, we found that heparan sulfate oligosaccharides are necessary to be present in close vicinity to the receptor, so that they can facilitate in the role of co-receptors the formation of the human interferon gamma receptor complex. I would like to acknowledge financial support from the Bulgarian National Science Fund under this grant, and computational access to computational resources here, and I would also like to thank you for your attention. Thank you.